All right. Next slide, please. Hi, and welcome to the next webinar in our Plantae webinar series. My name is Jason Padilla, and I'm your technical host for today's webinar. This webinar series is brought to you today by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. I would like to give a special thank you to all our ASPB members who are attending today's webinar. Your ASPB member dues help make support this webinar possible. For any of you who have not yet joined ASPB, you can join today using the discount code %10 to receive 10% off on your registration. ASPB webinar members get early access to these webinars, and you can learn more about ASPB and all the opportunities we provide at aspb.org. Next slide, please. You can view previously recorded webinars on our Plantae YouTube channel. There's so many other exciting talks. So if you're looking for great plant science content, you can follow our Plantae webinar, Plantae YouTube channel to tune in for more. Next slide. As you may know, the Plant Biology 2025 hosted by the Society, where many plant scientists gather to kind of share knowledge and commune with each other and kind of share network and all those things. Registration for it is now open, and this year it's taking place in Milwaukee, Wisconsin on July 26th to the 30th. Registration is open, and currently we're doing early bird, so you get pretty good discount if you register now till the deadline. It is a uniquely positioned kind of conference that brings together, again, members of the plant science community where you can acquire knowledge, network, skill, and learn new skills necessary to advance plant science and grow in your career. It is open to all career levels and also open to all just folks that are interested in plant science. To learn more, you can visit plantbiology.aspb.org. Next slide, please. Before we get started, I'd like to go over some housekeeping items. Pretty common, some of this for Zoom, but to make sure that you get the most out of your attendance for today's webinar. If you are experiencing technical issues, please let us know about those using the Q&A, or you can email me directly at jpadilla@aspb.org. If you have questions for our speaker, please add them in the Zoom Q&A section of your Zoom kind of control panel. Our moderators will be sharing them with our speaker during the Q&A. If you are trouble connecting or need to leave the webinar early, like our other webinars, this is recorded and will be posted on our YouTube channel. So again, if you are not following our YouTube channel, please go ahead and do that, and that way you can catch all the great contents that we offer through the Plantae platform and other ASPB spaces. And so without further ado, I'm going to turn it over to our Plantae Fellows, who will moderate and facilitate today's session. And so with that, take it away, Plantae Fellows.
Hi, everyone. Thank you so much, Jason, for that introduction and for running through all of our housekeeping items. So my name's Kira. I'm a postdoctoral researcher in starch in potato at the John Innes Centre in the UK. I'm going to be one of the three moderators alongside Carlos and Katerina today, who are two other Plantae Fellows. And we're going to be working with three speakers today, Marie Carmen Alamar, Anne Runkle, and Kati Ratzer. So thank you so much for all of our speakers for joining. Thank you everyone for joining in the audience as well. And I think it's a really great topic that we're here to discuss. So we just wanted to highlight different pathways to getting impact from your research. So a lot of the fundamental science that we do, it can be really hard to see how it can really apply within the food system, in nature. And yet it's never been more important. I think a lot of people right now especially are looking to engage a lot more with industry to get different pathways to funding projects as well, which can be really important for protecting your science and having different financial incomes for that. So I think it's going to be a great webinar, and I'm really excited. So first, we're going to have that general introduction. we'll introduce each of our three speakers, who will then present for about 10 minutes on their specific experience and how they kind of got impact out of their work. So throughout the session, there will be a Q&A available. So please put your questions in there as soon as you think of them. The speakers will be engaging within the chat as well. And then we will have a Q&A session at the end with all of the three speakers, where we discuss kind of more general questions, how you engaged, and then have a quick closing. And there will be a recording available at the end. So without further ado, I'm Kira, and I'd like to introduce our first speaker today, Marie-Carmen Alomar. She's a senior lecturer at Cranfield University who works in post-harvest biology of plants and how we can kind of have the best quality, maintain quality, get the best food for our consumers, prevent losses, which can have huge social, economic, and impacts on our environment. So without further ado, Marie-Carmen, thank you so much for joining. I'll let you share your screen.
Excellent. Thank you very much for the introduction. I'm now sharing my screen. So I hope that everyone can see the screen.
We can see that. Thank you.
Excellent. So again, thank you very much for inviting me. I'm going to give a couple of case studies basically about how post-harvest research can be translated into real-world applications. So one of the main challenges that we have nowadays is just to be able to provide the world's growing population with a sustainable, secure supply of safe, nutritious, and affordable, high-quality food. So normally effort has been made to grow more, have more yield, but that also incurs with kind of inputs in terms of more water, more labor. So we are, from what my research comes from, then we are thinking that perhaps in a short term, and just to try to avoid all the wastage of these inputs, natural resources that we have already put, is just we are looking at how we can reduce the food loss and waste that would happen once we have harvested those fresh produce. So the overarching frame, we are looking at the sustainable development goals, and that's how we normally frame and want to translate into impact the research that we do. So the first case study that I wanted to present here is around asparagus. So asparagus here in the UK, it's a crop that has very much importance within the economy. However, we cannot have a year-round supply of UK asparagus. For that, we rely on imports. However, these imports will come with some environmental impact in terms of CO2 production, because when they are sea fried, but also may have end up with some consumer satisfaction when there is a mismatch between what the consumers are expected and the quality of different produces. So that's okay. So what we will do just to preserve the quality of the asparagus crop is just normally using cold storage. But if we use cold storage alone, we only end up with 1 week plus 7 of cold storage, but 7 days of shelf life. In that case, what we have is sometimes that we end up with some disorders, and I'm putting here what we call a spear tip breakdown. So what happens is that the top of the spears, they start losing their integrity, they become soft, and also they start having a foul odor. So that will result in wider waste, not because the incidence of this disorder affects a high percentage of the crop, of the food that consumers will find in the retailer. Perhaps it's only 3-4% of the total crop. However, if we are thinking about, as a consumers, when you go to the retailer and you have a pannet, and then you have even one spear within the pannet, that may result into a waste of the whole pannet. That would happen at the retailer stage, and then you can even scale it up when you are thinking about importing batches of asparagus. and then not passing the Q&A. So in that case, then we want to find for sustainable solutions that can allow us just to extend that storage life whilst maintaining quality. So in our group, we have been researching and investigating whether dynamically controlled atmosphere could extend this storage life. And we did find that that was the case. So by reducing the levels of oxygen and increasing the levels of CO2 while spheres were stored at 1 degree Celsius, then we could extend the storage life up to 4 weeks. However, that disorder that I was mentioning about the deep breakdown still remain when we were passing spheres from 1 degree, which were the optimum storage conditions under the optimum CA conditions to shelf life. In that case, what we then came up with is that we really need to understand what are the mechanisms or the causes of this disorder, because we have a technology ready to be used, but the practitioners won't adopt it unless we understand the causes of the underlying issue. So for that, we continued our research, and then we had Innovate UK project where we were looking at the sphere tip breakdown from 2 different angles. From one side, are we able to identify spheres before they develop the symptoms? For that, we wanted to use non-destructive technology that would be using RGB cameras or also using visible and near-infrared spectrophotometer. So why do we want to identify? Because if we are able to see these spheres are susceptible, they haven't developed the disorder yet, but they are susceptible, then we can implement management decisions and then perhaps just put those batches upfront into the retailer, while the ones that are not susceptible and not having the symptoms in a while, then we can then keep them for the storage. So that was one of the approaches that we had. We found that with RGB cameras, we have a potential to be able to classify healthy and non-healthy spheres. But on the other extreme, we also wanted to understand the mechanisms, and on this front, we're still researching. We were having an approach where we trying to identify differential expressed genes between different cultivars, acceptable and not, that may be related with the tip breakdown. And also we wanted to understand the role that plant hormones will have in this disorder. So that part of the work has not been published yet, but it's a line of research that we are actively working on. And then another example that I wanted to talk to you about is about dormancy and sprouting in long-term storage crops. Here in the UK, potato and onions are very important crops. We have a high amounts of them that are being harvested, but most of them are also being stored for up to 11 months. So one of the issues that we have when they are being stored for that long period of time is that then we will have a dormancy break or a sprouting while in storage. And that will result in turn in a reduction of the quality and of course then into a food loss. So there are mechanisms that are being used. There are the use of different chemicals. There also the use of cold storage. CIPC was actually one of the chemicals that was being used here in Europe. 85% of the potatoes that were used for processing were treated with CIPC. However, in 2020, this chemical was banned because of the concerns on the human health. In that sense, then we wanted to investigate different alternatives, and actually we're working on this many years ago. And for example, the use of ethylene, we identified that they reduced the sprout elongation. So in that sense, that would be good because they will allow us just to get an alternative to the use of chemicals. And then further research also, we demonstrated that when we were using 1-MCP, which is an ethylene blocker that is widely used in other crops like apples, then we found, and we can see it on the graph at the top, that the sprout elongation in onions were further reduced in comparison with ethylene on its own. So here we have 2 avenues that we wanted to explore. First, why the ethylene plus 1-MCP was having that synergy. And also we wanted to try to improve some of the counter effects of the ethylene. I haven't mentioned it, but when we apply continuous ethylene application in potato, and now I'm moving to potato tubers, then the positive effect that we found, which is the reduction in the sprout elongation and therefore the extension in storage life, comes with a negative effect, and it's the increase in reducing sugars. Reducing sugars are a negative aspect when we are talking about processing potatoes, because when they are being processed and being fried, for example, of crisps, then that will result in a Maillard reaction and a discoloration. So in this case, what we wanted to see is whether this 1-MCP plus ethylene treatment will have, first of all, the same impact on the sprout elongation on potatoes, and then we did find similar things that what we found in onions. So in the graph saw that when we have 1-MCP and ethylene, we have that reduction in the sprout elongation. Then we moved and see, okay, what happens then with the reducing sugars? And we did find that the increase in reducing sugars that we will find if we are applying ethylene supplementation continuously was blocked, was inhibited by the fact that we were using 1-MCP. So we had a dual benefit. So we are able to maintain the reducing sugars very low. We are also able to maintain the sprout elongation reduced. And then when we were looking at the mechanisms, we identified that the ABA catabolism was being accelerated. So what is the result of this in terms of impact? So this is not only a research that ended up with a scientific paper, but a number of patents have also been filed out of this research. So it's how we can be able to not only have an academic output, but also have an industry impact that can be used widely. So now moving forward and trying to understand the mechanisms of that dormancy break, we did also some work where we were just assessing some diploid lines where the NSEED, the SEN1 gene was either being overexpressed or silenced. And we were able to identify that one thing is that what was the time to dormancy break, and something different was the time to sprout elongation. And in this case, we confirmed that the SIM1 was not actually governing endodormancy, but it did have an effect on the sprout elongation. We wanted to move this forward, and at this point, we are just have received some funding from the BBSRC for us to be able to uncover the rules of potato dormancy. So we are hoping that by integrating different disciplines and also academia and industry, so PepsiCo and Solinta being a breeding company, we would be able to untangle these two mechanisms, and therefore, at the end of the day, being able to perhaps govern and manipulate dormancy so that we can develop new cultivars that are more suitable, not only for those places where we would like to have longer dormancy, so that we can reduce the inputs during post-harvest storage, but also to adapt and having perhaps cultivars with lower dormancy for those countries where they don't have this need of keeping tubers for a long time. So this is a summary what I wanted to talk to you about, and I hope that you found this talk interesting. Thank you.
Thank you very much, Mari Carmen, for the great talk. And now we can move to the next speaker, that is Anne Rankel. So you can, stop sharing. Okay, and can you wait just 30 seconds that I present? Okay, so I'm another Plantae Fellow that I'm biotechnologies, doing my PhD at CBGP, that is a center of Madrid, Spain, and more or less my research focus on understanding the effects of high temperatures on plants and also on microbial recruitment. And I'm going to present the next speaker, that is Dr. Anne Rankel, that she's Vice President of Product Innovation at the Mosaic Company. She leads a dedicated team of scientists and engineers focused on developing science-based solutions for agriculture. Before joining Mosaic, Dr. Runkel head the research and development team at Plant Response, a startup acquired by the Mosaic Company. And you can see that she has a lot of expertise and experience on developing microbial solutions to apply to the real field, to the real world. So now, Anne, you can start with your talk.
Thank you, Carlos. Appreciate the invitation today. All right. Thank you very much for that introduction, and appreciate being invited to speak here today. So I'm an R&D leader who has worked in large and small companies focused on biological solutions for agriculture, primarily working on products that improve crop nutrition, quality, and yield. I have a PhD from the University of California, Berkeley in plant biology from the Plant and Microbial Biology Department. And I lead the product innovation team at the Mosaic Company today. Our team identifies, creates, and helps develop products for the Mosaic Company. The Mosaic Company, if you don't know it, is a Fortune 500 company with about 14,000 employees around the world. And we are the largest US producer of phosphate and potash fertilizer, and also production globally as well. So these are the P and K in your NPK fertilizer blends. In addition to developing new fertilizer technologies, our team also leads the effort to build biological solutions for the Mosaic Company, as Carlos mentioned, working on a lot of live microbe technologies. So today I'm going to share a few of my insights from my experience working in translating plant and microbial biology research into products that reach the market and ultimately impact crop productivity, impact growers, and the planet. So my core principles when working on product innovation and translating that research into real world applications are to start off by solving a measurable problem, then always doing good science, keeping it relevant, and collaborating. I'm going to walk through each of these with a couple of different examples. So when we talk about some of our live microbial technologies, one of the main areas that we're focused on is solving a really serious problem, which is that in the US alone, the combination of nitrogen, phosphate, and potash used as fertilizer is close to around 20 million metric tons. However, about half of that fertilizer that's applied is not utilized by the plant during the growing season. This is a really big problem, one that we're working to solve with some of our different live microbial products at Mosaic. And our product innovation team has developed new technologies from either individual microbes or consortia of microbes that help improve the utilization of those nutrients by crops. My next principle is always doing good science. And each of the products that we are working on started with solid scientific foundations and often come originally from some fundamental research. And often our technologies we work on started as partnerships with academic institutions or other companies. So bringing in new ideas and information from around the world to try to develop new products. In our research program, we're very intentional about our experimental designs and replication. So I'm showing some real data here from a product that we have developed. And data are messy. And so whenever we're working on developing a new assay or method, we, of course, as you all know, as many of you are plant scientists, there's lots of variation that can occur from one individual plant to the next, and then also how impactful the environment can be to those individual plants. So then when you add live microbial products like we're often doing for the things that we're developing, the system becomes even further complicated. Therefore, when we're working in the lab or working in the greenhouse or field research, we design our experiments with positional tracking, a high degree of replication for biological replicates, as well as full experimental replication. So the results that you can see on the left here are the combined data from the figure on the right. And you can imagine if you're looking at the run of experiment 1, 2, 3, or 4, that if someone looked only at the 4th run of that experiment, they would take away a different interpretation than if they had looked at 1, 2, or 3. So we're always very careful as we're setting up our experiments to make sure we have lots of replication, both across those biological replicates and across individual runs of the experiment. We also don't rely on one set of data. So this is looking at chlorophyll content. This is a greenhouse study that was done, but of course we're in parallel doing research in the field and across a number of different inputs to help us make decisions around how a product is working and how to improve it. So this is again clearly one of the most fundamental principles that we have is always doing good science and having science-based products. So the next principle is keeping it relevant. This is kind of a generic phrase, but what I mean by this is making sure that the product that we're developing and the science that it came from is actually going to apply to a real-world situation. So we need to turn that idea into reality, into a form that a grower would want to use in their field. So when we talk about our live microbial products, we develop those to be easy to apply for the grower. So they often can be applied right alongside other agricultural chemistries, including fertilizers or crop protection products. The products that we work on, we insist on having long shelf life, not requiring special storage conditions because those are not relevant for most growers. And then the other piece that's really critical in developing a product in an industry setting, of course, is the cost of goods. So I'm showing some cartoons here. You guys probably recognize the flask. Many of you probably recognize the little mini bioreactor cartoon, and then a larger scale fermentation tank. So keeping it relevant also means keeping a product affordable and cost effective for the grower. And when we're talking about live microbial products, some of these can be scaled up relatively easily and in a cost-efficient manner, while others require significant investment to reduce the cost of goods needed to make the product. And some are nothing is impossible, but some are truly outside of the range that is, at least at this moment in time, with the technology we have today, not going to be feasible to make it out into the field. Part of the reason for that is whenever we develop products, we expect that our products will bring a grower a 3-to-1 return on their investment. So what that means is if the grower is spending $10 per acre on the product, they should be getting $30 per acre in return. And that's often being measured in the yield return that they get on their field from that particular acre. And so whenever we're thinking about product development, again, keeping it relevant means making sure that it is usable, easily usable by the grower and cost efficient and gives them sufficient return for their investment. All right, finally, and most importantly, translating research into real-world solutions requires significant collaboration. Our project teams work cross-functionally, which means that we have representation from our R&D group or our product innovation team, as well as from commercial, marketing, operations, and even some other members of different functions within the company as well. Working with my cross-functional colleagues is really one of the most rewarding parts of my job. You recognize where you can contribute in an area, and then where you really need someone else's expertise to get something solved. So these cross-functional teams are also often where you recognize something serendipitous that a scientist alone perhaps would not have recognized as an opportunity, or a member of the sales team maybe wouldn't have recognized or identified a product as a developmental breakthrough. So this again is both foundational and required for good product development to have a collaborative attitude in the development of new products. All right. Thank you again for the invitation to speak today and talking a little bit about my perspective and principles around product innovation. I certainly don't claim to have all the answers, but these are a few of the key principles and have come from my learnings working in product innovation and translating that research into real world solutions. You can reach out to me here as my email at Mosaic, as well as my LinkedIn profile. I'm also happy to take questions if we have time, Carlos.
Okay, thank you so much. Thank you so much, Anne. Can you please stop sharing? And yeah, as if you have any questions so far for either our first speaker, Mari Carmen, or Anne, please type them in section. And let me introduce myself. I'm your last moderator today. My name is Katarina Kurtovic. I'm in a final year, a PhD student at the Charles University in Prague in Czech Republic. And I was, during my master's, I was studying plant hormone auxin from an agricultural perspective. And now I'm focusing on the evolution of auxin transport and its response in algal relatives of plant plants. And you can connect to me in the social media. And I would like to introduce to you our last speaker today, which is Kati Retzer. She's a principal investigator at the University of Natural Resources and Life Sciences in Vienna in Austria. She's leading a research in plant hormone signaling, root development, and plant microbe interactions. So she's leader of this large EU Horizon project called Barley Microbreed, where her team and she are focusing on development of resilient barley varieties through innovative approaches that integrate fundamental and applied science. And besides that, she's dedicated to mentor master students and PhD students, but also for fostering interdisciplinary learning. And you can connect with her on Blue Sky social media. From here, stage is yours. Kati, feel free to start your presentation.
Thank you very much for the kind introduction and also for having me today. So welcome again. I will today explain a little bit about one of the projects that I'm currently working with, and also a general interest of mine, by understanding how we can harness root microbiome interaction to increase plant fitness by taking the best out of the soil, by understanding how the genome environment interaction is interacting in the best way. So as Katharina mentioned, I'm at Boku University in Vienna, and I'm one of the coordinating partners in the EU project Barley Microbreed. Yeah, so basically, since 15 years, I'm interested in dissecting and understanding how roots interact to external stimuli, and also how this changes the root function and also the way how they spread through the soil. So when you work with roots, the hidden half, our biggest problem is that when we are working in field conditions, then of course the roots are in soil, so they're hidden from us. But on the other hand, when we're in the lab, we need clever ways to study roots, because otherwise we can influence how the experiments are giving us the results. So one big part of my personal interest is, since several years, to improve the way how we cultivate roots for experiments, and to develop also novel tools to get physiologically relevant results. Yeah, so in the Barley Microbreed project, which was funded 22 For 6 years, we are interested to understand how roots interact with indigenous soil. That means that we are observing the plant yield, but also the root architecture and function of 568 barley varieties in 5 yields in 3 countries. So we have a field in Austria, of course, Morocco, in Lebanon. We have always 2 fields. And basically, root architecture is something that is easily accessible. So the first part is to understand if there is a difference in root growth patterns depending on the environment. We have the experiments running, but on the other hand, we also have to focus on root function. For example, root function is the ability to secrete root exudates, which is attracting the microbiome. And by observing if there is a high or low rhizosheet formation, we have also a first insight if the plant has good availability, depending on the genome, to interact with the local microbiome. So why is it important to study root microbiome interactions? Of course, a plant is not always able to get the nutrients that it needs from the soil when it's bound in non-soluble forms. So the microbiomes can break down those nutrients like nitrogen or phosphate, but it can also enhance water conductivity, which is especially important in very dry regions. And of course, it's a safety shield against pathogens. If you have like a nice rhizosheet, pathogens cannot easily penetrate the root. So to understand how roots interact with the local microbiome can improve our understanding which varieties to use in which areas, and this could result into higher yields because less water is needed in drier regions, less fertilizer and less pesticide usage. So just to see, so the project is led by our colleagues in Aarhus, Mette and Mons, and then together with ICADA, which is a breeder association in Morocco and Lebanon. They are specialized to select especially cereals and legumes for dry regions. And with my team at Boku, we try to coordinate this project, but of course we have a lot of other great colleagues. I have here an overview slide. We have a holistic approach. It's a field to lab. It's as I know, the biggest in-field to lab approach at the moment available. We try to understand the plant genetics depending with the microbial ecology. And then we also include soil science. And then we try to find the right QTLs. And we have great colleagues at Aarhus University and Copenhagen University trying to link this, QTLs with root phenotype and microbiome association, and of course yield at the end. This is just that you have an overview about our field sites. We have from European sites that are well rain-fed to very dry regions in Morocco, different environmental situation that we can compare with each other. And after 2 years of field studies, we have quite a good idea which of those almost 600 genotypes have which impact on interacting with the local microbiome. And then sometimes we have the situation that we have a bigger influence of the environment, and then sometimes we have a bigger influence of the genotype. And then of course it's important in communication with the local farmers and also breeders in Europe. We try to understand what are the needs and in which way at the end of the project we aim to, when you see here in Work Package 11, to decide which breeding strategies will be the best So we will take all our basic knowledge, translate it according to the needs of our collaborators and breeders and farmers that we are constantly exchanging with. Yeah, so if you want to know more, please check some YouTube videos we have on our Bali Microbreed channel, and then you will learn more about the project and our holistic approach. So as I said, it's not easy to work with roots when you are observing them in field. They are like a soil is very heterogeneous. You have always to have a good idea which kind of soil you're working with. The total environment can change very drastically, like climate is changing. Water availability is always different. So it's very important to have a plastic plant that can adapt, and to understand how to achieve this is one of our main goals. And of course, there is like the interest of the breeders, biotech firms, and policymakers that are involved also. And we have a lot of dissemination projects where we always try to get some feedback from them during our project. So of course we have long-term goals. This is like to develop like the so-called next generation barley varieties that adapt best to climate change, require fewer chemical inputs, which is also better for the environment, and to maintain by this also the soil biodiversity. As I said, one of my biggest personal interest is to improve also in lab root phenotyping. As I said, roots are very sensitive. They react to many stimuli at the same time. And just to get the best results during our experiments, we developed a tool that you can assemble by yourself. It costs, one of these boxes costs below 200 euros when you assemble it by themselves, but you're able to track via infrared lamps root growth directly on soil in darkness. And you can try different treatments, you can try different soils, and you don't have like the toxic effect of direct illumination on the roots, which was shown in several studies to have a negative impact on experimental results. So if you are interested, please check out our preprint from last year or contact us, and we are very happy. So let's see if the video will start from the beginning. Yeah, so this is how it looks like. So we can track from the beginning of germination exactly the growth speed, different root rates at root angle growth, root diameter, and other traits very easily, and get a dynamic picture of individual genotypes depending on the soil we are using in the experiment. So in terms of image analysis, we can easily, by using, for example, Riser Vision Explorer or other programs, grab the information from individual pictures and get very dynamic data exactly at what kind of experimental setup you have. And of course, we are not happy with this yet, so we want to develop like a infrared-based microscope. We are at the moment using a 4 times and 10 times objective, because, as I said, most of the interactions and actions of the root happen on cellular level. So we want to be able to, under the most controllable conditions, track those responses, for example, like root hair formation, root hair elongation speed, but also formation on rhizome sheet, and to measure rhizome sheet diameter, for example. But at the same time, it's very easy to sample the rhizome sheet and root exudates in the setup for downstream analytical methodology. So basically, we are not preparing a manuscript, but you are always welcome to contact us beforehand. So this cost of one unit is the same, like around 200 euros. So we want to keep it for everyone available. And when you have a question how to set it up, it's like a 3D printed microscope with a movable stage with ring light illumination to where you can adjust infrared lamps to get a nice image of high resolution, and easily track the whole route up and down and get a good view on what is going on, depending on the soil you're using or the treatment that you're using. So basically, the key takeaways of this talk is that root hairs seem to be very central to my plant microbe communication. And in our project, the Bale microbreed, we want to highlight the potential of different root traits and to optimize and make people aware that root traits can also support whole plant fitness in a way that it could be interesting for breeders and also downstreams for farmers. So of course, our next 4 years are focused around selecting breeding material, enhancing drought resilience, push towards innovative breeding, then go back to the field, validate our findings, and of course, deliver non-destructive root phenotyping tools for the lab environment to confirm the data that we found in the field, and engage more with industry and policy partners. And by this, I would like to thank everyone who is involved in the project, and also all my colleagues at the Institute and at BOKU, and especially my team. And I'm open for your questions.
Thank you so much, Kathy. And I would like to, in the end, thank to all three speakers for their incredible talks. And with this, I would like to start the Q&A session. If you have some questions, feel free to type them down. First question, this is a general question for all three speakers. In mixed industry and academia projects, how do you decide what information will be publicly shared? For example, in a shared published paper.
So perhaps I can, if I may. This is always a conversation that we start even before actually starting the project itself. So from project development, we have to be very aware about what our outputs would be and what the expectations of each of the partners are. So as academics, we are committed to science. We even some fundings with they require that the data is publicly shared, because that's part of the funding guidelines. But on the other side, and we have IP, and companies would have also invested and have some sort of protection on IP. So from my experience, that's something that being open and being collaborative can be resolved, but a conversation could happen from, as I said, while we are developing the project. So of course, when it comes to what has to be published, we will need to go through all the clearance processes from the different companies, because sometimes you are not only collaborating with one company, so it's something that everyone has to be comfortable with. But at the end of the day, we find ways by which we are able to publish. So the example that I was providing before about the work on a potato sprout elongation and so on. So in that case, what happened is that the publication was somehow a bit delayed in terms of being submitted to the journal, because we were waiting for the patents to be filed. So it's always a bit of a compromise, because it may delay a bit things, but if there's a will, then it's a win-win situation, because you have both the impact in the scientific community, but also the industry and society impact.
Yeah, I would just add that from the industry standpoint, it's actually very beneficial to have publications about your products or technologies that are out there and vetted by the scientific community. So, of course, it depends on the project and exactly what is being shared, but oftentimes an industry, we're very excited to have publications and collaborations with academic institutions.
Yeah, so I would like only add on, it's the same in terms of the EU project, that of course first you decide all together as a team who can share what. Of course, like our breeders have to think a bit more carefully what they share, but you also have to announce your deliverables if they are open to publishing or not. And of course, it's better in terms when you use tax money, when you promise to have available deliverables, and this can be publications, reports, or other kind of information that you share on the platform or as publishes manuscripts.
Okay, thank you so much. Now we have specific questions for Dr. Runkel, but maybe also for other speakers. How do you choose relevant research direction in a fast-moving industry?
Yeah, that's a great question. So we set fundamental goals, of course, across 5-year plans as well as within the year. And then we have a clear strategy around the products that we're trying to develop. So we don't necessarily know exactly what that product is going to be, but we have some very clear ideas about what it could be and structuring those across a full timeline. Now, as you said, it's fast moving. Things change, but we do set up pretty clear strategies in advance in terms of what kinds of technologies we're working on. In the case of, some of the things that we're working on today that I was talking about, we have a big picture goal around nutrient enhancement and crop nutrition, and look at a variety of different technologies as they come out throughout the year and incorporate those into our plans.
Thank you so much. Now a question for Marie Carmen, and then again, it's relevant for maybe all 3 speakers. How machine learning and AI can be used in post-harvest research, especially in your omics studies and in the food system to reduce waste?
Okay, so it actually can be used at many stages. So in the post-harvest side, for example, when we are talking, I gave an example about spears, asparagus spears, where we don't know, if we are using human eyes, because sometimes when you are in a sorting line, you will sort the crop or the fruits by size, by color, but sometimes, you need a huge amount of information. If you are doing it with humans, that can be subjective. So at that point, you can develop that predictive models, either just to classify based on quality. You can classify or predict disease development. So at that point is very important. And we are talking at the very end of the point, so when crop have already been stored, and just before being able to being packaged, then you can get that information. All of that will allow you just to have different tiers of quality. You can have premium and non-premium. So that's a point where it's definitely being used. And at the other extreme, where AI and machine learning can be used, is, for example, at the breeding stages. So we collaborate very much with breeders, with breeding companies, and then when you are looking for a trait, then you need to phenotype a large amount of lines or genotypes. That is something that normally is done manually. That's time consuming, and again, it's fatigue, it's subjective because it would depend on whoever is doing it. So if we are able to have a robust AI or machine learning model that is able to phenotype and have a throughput way of doing it, that can speed up all that at that part of the breeding process, which is pretty much valuable. So actually you can use these tools at different points of the supply chain, straight from breeding programs during cold storage, because then you can monitor. It doesn't have to be only imaging. You can monitor gases, or you can monitor other traits, but you will still need to use AI or machine learning models to look at the data. So it's pretty much an area that is developing and expanding quite strongly. But that said, you need a good amount of data and quality data. So many times you say, oh, you apply an AI, I've got a model, but you have to have something that is robust and you are able to apply it confidently when the different traits that you are measuring. So a lot of effort put into that front as well.
Thank you so much. Now to finish the, not finish, but get the full circle, I want to ask a specific question to Kati. How do you quantify the root traits in the field, and how can these be integrated in the breeding strategies?
Yeah, so it's highly laborious. So you have to dig out the roots. So it's a bit different, like remote controlling and evaluating of shoots, where you can use drones or satellites. Meanwhile, we have really to dig out every single root you want to examine. So there are several ways. So either you focus on the topsoil area. The technique is called shovelomics. So you dig out the upper part of the root, and then basically you either take images directly in the field, or you bring it to the lab, wash it, scan it to get a better resolution. And depending on how many roots you have, you can also apply machine learning supported approaches. And also some programs that are already developed, like, as I mentioned, the RisoVision, that it's open to everyone to use. You can also go deeper into the soil levels, like by coring. So you take like soil cores down to the depth that you need, and then you also have to wash. So everything takes a lot of effort. So the limitation is the workforce you have, and also the time. So you need much more time to phenotype roots in the field compared nowadays shoots. What is beneficial, of course, when you know how a certain variety, certain plant is adjusting root architecture in your local soil, then you know you have, for example, a root that is growing in the topsoil or is growing towards groundwater. That, you know, if you're a farmer, you want to know, if I'm in a rainy region, I want a root that has topsoil roots, or I have a variety that is reaching very fast to groundwater when I'm in a region when it's not raining so often. So this is how you can use architectural traits to assess which varieties are best for you. And on the other hand, root function, for example, as I mentioned, root exudation, we have now several varieties that have different root plasticity, and those behave differently, like in Austria or in Morocco, where they cannot. So when you don't share carbohydrates with the microbiome will be not interested to interact with you. So so there are different parameters we know we can measure. We're learning during the project more how we can use those parameters, but basically that's how at the moment it's looking like. So it's a lot of work, but I think it's worth it.
Thank you so much. And last question for all three, last quick question. What advice would you give to PhD student who wants to transition into industry, especially if moving to a new country or culture?
I'll just start quickly and say one of the things that was very helpful for me was to partner up with some of the industry academic partnership alliances through the university program that I was at the University of California, Berkeley, visiting different companies if they have the ability to do that. ask questions, reach out to people like me and chat and kind of understand what it looks like and what are some of the things that you could bring to an industry environment.
Okay, if that's would you like to say something, Kathy, in the end, or Nadine?
No, I just wanted to agree. Yeah, that's yeah.
Yeah. Okay, for this, I would like to end our Q&A session. If there is anything you would like to ask, you feel free to reach out to the speakers directly, and I'll head over to Carlos.
Yeah, I'm just going to wrap up. So thank you to our incredible speakers also for accepting our invitation and for sharing their insights, and also all the audience who attend. And I also remember that you will have this webinar recorded in YouTube, just if anyone wants to check. And we hope that today's discussion has provided valuable insights and inspiration for your own work. And thanks again to everyone.
Thanks, everyone. We hope to see you in the next one. And a big shout out to our speakers and to our Plantae Fellows who moderated today's webinar, and of course to all our attendees who are very engaged and ask great questions. Thanks, everyone, and have a great weekend.
Thank you very much.
Thank you. Bye.
Bye.
Thank you, everyone. Bye.